Hello everyone, Peter Newman with you. In AHRI this week there's a bit of a buzz around the corridors because they've just had a paper received into a very prestigious journal, the PNAS journal, the Proceedings from the National Academy of Sciences in the USA. Now the reason they achieved that, it's the work of Lang Pan, a Chinese student, and Yuxin and others from AHRI, is that they found a novel glyphosate resistance mechanism, another world first resistance mechanism, and that resistance mechanism is very similar to multi-drug resistance in cancer. It's the same mechanism. So let's take a look at what it is. So we've got a plant cell here. This is the plasma membrane, the cell membrane holding the whole cell together. The resistance mechanism they found is related to these ABCC transporters. So plants have a lot of these ABC transporters on membranes around the cell, and they particularly found that it was the upregulation or the overexpression of this particular ABCC transporter. Now what happens is glyphosate can diffuse into the cell, high concentration outside the cell, low concentration inside, simple diffusion, but when it gets in there it dissociates into its anions and it gets trapped there. So the only way glyphosate could get out of that cell or move around is with active transport. What they found was these ABCC transporters, the resistant plants had more copies of the gene, therefore more of these on the cell membrane. These transporters grab hold of a glyphosate molecule and then they change shape like this and push the glyphosate, pump it out of the cell from the cytoplasm into the apoplast. Now the apoplast is essentially the space around the cells, and glyphosate has no activity there. So glyphosate is trying to get to the plastids to bind to the EPSPS enzyme. By pumping it out of the cell, we just reduce the concentration in the cytoplasm and reduce its efficacy. Now, as I said, this is a very similar mechanism that what happens when they treat cancer patients. They can be treating a patient with chemotherapy. The tumour seems to be shrinking and disappearing, all's looking good, and then the tumour comes back. And one of the mechanisms is exactly this. The chemotherapy drugs are pumped out of the cancer cells with ABCC transporters on the membranes, and therefore the new cells that then divide and become the new tumour have lots of these transporters, and there's multi-drug resistance, so it can resist many different chemotherapy treatments. So really interesting and exciting when we have scientists working in disparate fields, weeds, cancer, and their research overlaps. So hopefully they can learn a little from each other. And just while I've got this cell on the blackboard, I thought I'd go through all of the known glyphosate resistant mechanisms, just a quick overview. So we have glyphosate diffusing into the cell. It's trying to get to the plastids where the EPSPS enzyme is. What other ways that a weed or a plant can resist glyphosate? So one is this mechanism we've just spoken about, ABCC transporters on the cell membrane, grabbing hold of the glyphosate, pumping it out of the cell into the apoplast. That's one. There's another similar mechanism, which is vacuole sequestration, where we have similar sort of transporters. This membrane around the vacuole is called the tonoplast. We have tonoplast transporters in that membrane. They can grab hold of glyphosate and pump it into the vacuole where it has no activity. Also, at the target site, the EPSPS enzyme, we have the 102 and the 106 mutation. So the 106 mutation on its own giving lowish level resistance, but the combination of the 102 and the 106 mutation giving the highest level, sometimes 100-fold glyphosate resistance, the highest level of glyphosate resistance we've seen. Another one is called gene amplification. That is where we have the genes for the EPSPS enzyme. We have plants evolved to have many copies, maybe 100 times the copies of those genes, so that we produce lots and lots of EPSPS enzyme, and it's just normal EPSPS, there's no target site mutation, and the glyphosate can still bind to it and inactivate it, but there's so much of it, even though the glyphosate inactivates some of that enzyme, there's enough left over for the plant to do its normal purposes in the cell. And a final one, giant ragweed has its own, a weed in the US has its own mechanism. If we have a plant growing with its leaves off the plant, the treated leaves, they fall off really quickly. So the plant has this rapid tissue death. The treated leaves die very quickly and fall off, and that stops the plant taking up and translocating a lot of glyphosate, and then the plant is able to recover. So there are lots of glyphosate resistance mechanisms, all the subject of lots of work at AHRI and other research institutes all around the world. I'm sure there'll be more in the future, unfortunately, but just amazing to see just how adaptive plants are and the science that's gone into discovering all of these resistance mechanisms.